It's a very large cardiovascular award, which is stewarded by McGill and was initiated in 1978, following the endowment of funds by a lady, Dame Libovici, in honor of her two brothers, Arthur and Louis. And on an annual basis, the committee has had the opportunity to review nominees from all over the globe, and had always the pleasurable task of choosing some extraordinary cardiovascular researcher to receive the prize. So this is my opportunity, excuse me, to present Dr. David Kass, who won in 2020. Now, normally he would have visited in the year following the award, but because of COVID, this had to be deferred, as he was anxious to make an in-person visit. So it's a little bit frustrating for us all that it's still, some of it is still virtual. However, there will be in-person meetings with a number of groups across the campus in the coming days, and hopefully we will not exhaust him. So Dr. Kass is currently Abram and Virginia Weiss Professor of Cardiology and Professor of Medicine at the Johns Hopkins University School of Medicine. And he graduated with a BA in Applied Physics and Engineering from Harvard College, and subsequently took his MD from Yale University. Then he did his training in cardiology at Johns Hopkins University, where he has stayed ever since. He's been the recipient of a number of awards in addition to this, to the Lucian Award. He has a staggering publication record with more than 500 publications and an H-index of 141, probably rising as we speak. He's a world leader in a variety of topics that relate to heart failure, cardiac physiology and mechanics, second signaling in particular in relationship to protein kinase G and phosphodiesterase. He's also been involved in pioneering the development of cardiac resynchronization therapy. And of course, many of his studies are achieving application in the clinical context. So it's a great pleasure to have him here, and I would invite him now to give his presentation. Thank you very much.
Thank you very much, Jim, for those kind words, and it's a real honor to accept, be here for the Lucian Award. My name will go down, I guess, with an asterisk in the list, as delayed due to COVID. Although I guess maybe this past year, maybe I don't know, it's a year after, so maybe the next one actually got to come right away. So I'm going to talk about a passion of mine that started quite a few decades ago. And it's now quite a topic of conversation, which is heart failure preserved ejection fraction. And I said on this slide, no disclosures. I lied, there are some disclosures. But none of it's really going to be all particularly relevant. But to be compulsive, these are things that I'm doing that have something whatsoever to do with the HFpEF. And I was asked for some learning objectives, so I'm going to go through some learning objectives. If it'll, well, I see what I have to do. So yeah, I'm going to try to go through what is really a multifactorial nature of HFpEF, learn about some new cellular molecular insights. This is work that we're really very, very much pursuing right now. And then look at existing therapy, ongoing trials, and some future directions for therapy. So to start with, just to get a scope, what is HFpEF and where does it fit? This is the scope of heart failure generally. And these are just numbers. So this happens to be the total number of patients in the United States who have heart failure. This happens to be the number of new patients. Is there a problem?
Yes, apparently the screen is being picked up by the camera and the slides are not showing as they should be. So we have to do some adjustments.
Okay, so I'll definitely do it before we get too far. So, no problem.
I'll log on to my Zoom account.
So this room can see it, but no one else can see it.
Goodness.
That would be.
We're here.
Y'all welcome.
Do you have Zoom on?
Come on down.
Do you have Zoom on your computer?
Pardon?
Do you have Zoom on your computer?
I do have Zoom on my computer.
I'm not very good.
No, Zoom.
Oh, it's there. I will connect on my.
The magic numbers.
I'm not sure if it's gonna work. I don't think it's supposed to be. I share it with him as well.
I may not be on the network.
I know the new to network.
Right. So in order for me to do it, again, is it easier still? Unless I don't know how to connect to you guys.
I won't go public.
Okay, now I'm connected. Now it's connected. Hopefully, since I've joined a new network. No, it doesn't want to connect.
But the whole Wi-Fi usually is a pop-up, but you have to put like a user password and stuff. Well, it's not in Wi-Fi.
Well, again, I have it on a pen drive, so it may be faster if everyone is otherwise. Because I didn't ask me for anything. And it's not, it doesn't want to join your network. For all of you out there who are waiting, don't go away.
It's because it's so far away that I don't see your slides.
So far away. Oh, the camera? Oh, I see. That's no good. That's no good. We'll try Zoom again, but if this doesn't work, then let me go get my pen drive. We can just plug it into your computer.
Yeah, that's what I'm gonna do. Because I'm already connected on my.
Do I get extra time? Sure. I'll talk even faster.
That's what you can actually do. Try to play some Safari because there should be a pop-up that you should be able to connect to the Wi-Fi. So there's Safari.
No, we'll just put it on the computer.
Yeah, let's just put it on the computer because at this point, it's going to be probably.
Okay.
It's right there. How do we remove it?
We go to commercial.
Kingston, this one?
Yeah,
Montreal,
Montreal
GR.
Grand Rounds, yeah.
discussion.
Second, the first screen as well.
So we're already in the meeting. Share screen. Your presentation. Do you want to start from the beginning?
Well, yeah, I'll scroll, but
Yeah, turn the screen one.
Yeah.
We're still just seeing, we're just seeing the camera view still.
No, we're gonna share the screen. But where is it? I don't see it anymore. Yes.
We can see it remotely now.
Please move this window away from the share. And then desktop.
Desktop one.
No, I can't see his presentation. Let me just.
see it now?
No, we still have the camera view.
Where's my moving? Okay.
I'm going to go to my office and see if I can hit it.
We can see this remotely,
So you see the full screen, right?
almost.
Almost.
Because they should.
It's not in presentation format, but it's fine.
Can we get rid of the individual postage stamps to the right?
What I want them to see is not the panel, it's actually is your presentation. You guys don't see the presentation? You guys see this?
Perfect now.
So this is Zoom, this is what they see now.
And I can do that. And it shouldn't change anything. Good? Is it a resounding yes?
Yes.
Yes. So I'm back to the scope of the problem. We're good. Okay. Sorry about that. Okay. So as I was saying, we have 6 to 7 million heart failure patients just in the United States alone. We have 650,000 new people showing up with this diagnosis on an annual basis. Worldwide, there are about 64.3 million patients with heart failure. This is not an orphan disease. We spend roughly $350 billion to take care of this 64 million patient population, so it's not an inexpensive problem. And its outcomes still aren't so great. This is still about the 5-year mortality rate, which is 42% for heart failure. This is the 5-year mortality rate for stage 3 colon cancer, which actually is a little better. It's a serious problem and affects a lot of people. And half of all of that is this. Half of all of that is heart failure, where the ejection fraction basically looks okay. So we go to the book of HFpEF for the historic perspective. Here's a case presentation. This would have been what I was seeing when I got interested in this a long time ago. 64-year-old female with a history of hypertension, atypical chest pain, type 2 diabetes for a while, hospitalized for dyspnea with pulmonary edema. Started, yeah, well, that was not, that's more recent. In the emergency room, is very short of breath, hypertensive, afebrile, ejection fraction on echo is 65%, left ventricular hypertrophy, valves look normal, no evidence of ischemic heart disease. She responds to diuretics very, very quickly. And you see her again in your clinic, and really her primary complaint is that she's easily fatigued. This was very classic. So what is this? This is what Eric Topol, actually, back in 1985, when he was just a year ahead of me in my fellowship, had published in the New England Journal, hypertensive hypertrophic cardiomyopathy of the elderly. We used to see this all the time. We called it LOL for little old lady before I laughed out loud became popular. But essentially, these were hyperdynamic ventricles that were cavity obliterating. And this was a hyperdynamic form of disease that presented with symptoms indicative of heart failure. But it looked like too much systole. And then, equally strong in terms of explaining this was that they were supposed to be stiff hearts because of hypertrophy. And so there was not enough diastole, they couldn't fill. So these small cavities with a hyperdynamic, hypertrophic, hypertensive cardiomyopathy. Now there are a variety, this was a cartoon. The figure on this side actually shows some data from myocardial strips from HFpEF patients published much more recently from Mike Zeil. And these were the group that were suggested to be hypertensive HFpEF patients as opposed to the controls down here. And clearly there's diastolic stiffening, higher stress for the same circumstance. So we tried a lot of stuff. Basically, we started the stuff we use for HFrEF, right? So we had ACE inhibitors and ARBs and aldosterone antagonists, and we even tried Viagra. We tried nitrates. We tried beta-adrenergic receptor blockade. And the bottom line was basically it didn't work. None of this worked. And this has really been, as a result, one of the largest unmet medical needs, certainly in cardiovascular disease, but arguably in a lot of medicine, because it's a big problem. And yet we really had, till very recently, we had no evidence-based, approved medications. So part of the issue when things like this are going on is you sort of step back and try to figure out, what are we getting wrong? Part of it was that it wasn't just what we thought it was. There were lots of other things that were going on. This was one of the studies that Barry Borlaug and I did in the mid-2000s, where we took a bunch of HFpEF patients, and the control group, and this is an important control, were not normal people. They were also matched. They were like case matched, so that they had hypertension, LVH, they had obesity, they were on the same drugs because they had hypertension, so they're also on calcium channel blockers and beta blockers and stuff like that. They just didn't have heart failure. No one ever said they had heart failure. No one ever hospitalized for heart failure. That's our control group. And this was one of the mechanisms that we identified, which is it's not just diastole, but they can't get their heart rates to go faster when they exercise, which is a normal chronotropic response that contributes to improved cardiac output. And they also couldn't decay their heart rates as fast right after they stopped exercising, which had been around this time, just a little bit before, had been already identified as a risk factor actually for adverse cardiovascular outcomes and death. So there was something clearly wrong, neuro control system that was regulating heart rate as a component. The other thing that we found was that this correlated to their peak oxygen consumption.
Somebody in the studio audience wants your microphone off, so you speak only into the camera, into the computer.
Okay.
I don't know how to turn it off, so I'm just going to put it away.
Is there an echo? Is that the
Yeah.
reason? Great. Okay. Sorry about that. Whoever's in the audience, hopefully it's better. If it's not, let us know. So peak oxygen consumption correlated to this rate of heart rate change, and this is a very important determinant of net exercise capacity. But also what we were finding was it was in the periphery. Their muscles were not dilating, the blood vessels were not dilating. Even if the heart could put out more cardiac output, the tissue that needed it to exercise wasn't basically receiving it. And this was something we had known about in HFrEF. So these were already suggesting there was more going on. In terms of skeletal muscle, it is true, lots of fat in beef is tasty. But it actually isn't really good for you. And if it happens in your own muscle, it's particularly bad for you because it impedes normal metabolic function. Glucose resistance goes up. There are myokines that get secreted in skeletal muscle that has fat. And this is actually an MR image of fat in the thigh muscle of a HFpEF subject compared to a healthy control subject. And all this marbling that was in our Kobe beef is causing actually major metabolic problems in these patients. So this is not even the heart now, and now we're moving out. So my vision of this disease for some time has been the straw that breaks the camel's back disease. We've got all these different inputs, some of them heart, some of them not. Pulmonary hypertension, obesity, diabetes, metabolic syndrome, muscle sarcopenia, sleep apnea, chronic obstructive pulmonary disease. These are all kind of colluding in a not particularly great way to create this complicated systems disease that is HFpEF. And one of the advantages of this analogy is that it doesn't tell you what straw you have to take off to, so the camel's back is better. There's nothing, even though we call it HFpEF, it doesn't have to be a heart target. You could do other things. You could go after some of these other things, and some of them may indeed be more tractable. I think that's partially what we're seeing now, finally, with some of the new therapies. So when I asked the question in the first slide, how has it changed? This is sort of how it's changed. In the meantime, while this was evolving, we were doing all these negative trials. The world was getting heavier, and it's not because there are more of us. It's basically because we're heavier. And I know when I looked at this last night, that Canada always remains a lighter color of red than the United States. We're ahead of you on this one. But basically, we've got a rising rate of obesity. In the US, it's 40% of the population with a BMI over 30, 20% exceeding 35. It's a major comorbidity for so many diseases, including cardiovascular, but also cancer. And small molecule therapy for obesity is still lacking. I mean, we have bariatric surgery, and we don't have that many things to deal with, particularly this level of obesity. And what I'm going to try to convince you of is that this part of the equation that really didn't exist when I was doing my studies in the 80s and 90s, but has definitely now taken over as the major comorbidity, and certainly 2022, is really changing everything. And it's changing, I think, underlying pathophysiology. So this is HFpEF in Baltimore in 2022. Our population is about 60% female. We are, as an inner city population, 68% are African American. Hypertension is more common in African Americans, and hypertension with hypertrophy is more common in African Americans. About our median BMI is 41. Our 25th percentile is 36. So we don't have very many people, right, who are not obese. Unusual relative to the clinical trials, we take care of a sick population. Kavita Sharma has established a HFpEF clinic at Hopkins. It's one of the only 2 in the country that really focuses on this syndrome. And the majority of our patients are class actually 3 or 4, not even class 2. So why has this been so hard to come up with something that works? Part of our problem is that with that straw that breaks the camel's back analogy, you can appreciate this is not something you can easily do with a mouse or a dog or cat or anything else you want, because you've got all these kind of comorbidities. And how do you start selecting? Scientists are really great at reducing things down to, I'm going to create pressure overload, or I'm going to give dope of salts, or I'm going to make an infarct, or these are nice models. This disease is none of that. This disease is like a big gamish. And I think that's part of its problem. And it occurs in humans later in life. I mean, you've got decades of accumulated comorbidities, and this is not tractable to do in animal models and labs. So we really have not had good animal models. We have some better ones now, and we're making contributions there. I'll talk about that a bit more on Friday, but nonetheless, this has been an impediment. And the other thing is, scientists love to reduce things to cells in a dish. You cannot put HFpEF in a dish. Sorry, it's not going to happen. And so even, you can play around with all your different combo organoids, but I don't think we're going to be able to really create it there. So to some extent, that's what's driven, it's what got me out of this field. When I started by somewhere in the early 2000s, I couldn't figure out what else to do. My lab is basic science. My clinical stuff, we had done almost everything we could do. I didn't have a cohort, I didn't have a clinic like Dr. Sharma has set up. That all changed when Kavita came somewhere around 2013, and suddenly we have a coordinated center. And for one thing, she's a heart failure physician. She biopsies. At Hopkins, we get into cardio-biopsies. It's a tradition at our place. It's very unique. So I had access now to human myocardium for the first time in a large population of patients. That's, for me, it's changed everything. And so I'm going to show you mostly the data that's come out of that work just in the last couple of years and where we're going with it. Because I think it's really pivoting our conception of what this is a disease is about. And even though all those other straws are on the camel, I don't have biopsies yet of their visceral fat. We're trying to get skeletal muscle. I'd love to have their livers. I'd love to have, but we got heart. So you're gonna learn about heart. That's what I can tell you. Now, one caveat to what I'm going to show you is that these are all right heart biopsies because that's where we get our tissue. In Europe, and both in Belgium and in Germany, there are a couple of groups that are doing left heart, and there was some limited data from left hearts. That's all from Europe. Big issue with that data set. It's about 20 years old, so it's actually more old HFpEF. Most of these patients were very hypertensive and had LVH, mostly male. Not what we see now, and almost no one with obesity. So you have to take that with a grain of salt. But what I'm going to show you is basically reflective of what we're seeing in Baltimore. And I wanted to make the point that even though we're getting the RV is important, actually, and RV dysfunction is increasingly common in patients with HFpEF. And these are just a series of published studies over the years. And the percent of patients who have evidence of right ventricular disease is not insubstantial. And this is what we see. I'm going to make a point a little later in this talk that there's a lot going on at the basic level that we don't actually see, that is certainly not embodied by ejection fractions and wall motion and things that we're used to looking at. But it is what's going on inside the sarcomere. And also importantly, when you have RV dysfunction and HFpEF, your mortality rate is much greater. So figuring out what's going on, even if it's from the RV, is going to be very important. And it's certainly, I think, a intellectual driver and medical driver for this enterprise. So Virginia Hahn, who first did a Sarnoff fellowship with me when she was a medical student, and then came back to Hopkins as a fellow, published this paper in 2020. And it was really the first stuff coming out of this RV histopathology of HFpEF. And it already suggested some things that are different from what we thought. Because we thought these patients were all going to have lots of hypertrophy. And another popular idea is that they get a lot of fibrosis, and that's partly why their hearts are stiffer. That's why they have diastolic dysfunction. Well, they do have fibrosis, and this was all done histologically from basically, I think, a little over 100 individual patient biopsies. But you can see what's shown here is that the vast majority, it's really very mild. And only a few of them, a small percent, have moderate, and very few actually have severe fibrosis. So it's there. But is this the driver of the entire thing? It's kind of hard to imagine that when so many of these patients, it's really mild, and it's really not that different from control levels, and I'll show that in another slide. Hypertrophy, similarly, was moderate to severe in about half the patients. So a lot of these patients really don't even have much hypertrophy. And you're seeing that more and more in clinical trials that are getting published with newer drugs. The level of LVH, instead of being 80% of the patients, is now down to sometimes less than 20% of the patients. So our mindset is that this is still hypertrophic heart disease, I think is wrong. The other interesting thing was that there was undiagnosed amyloid disease in approximately 15% of these patients. Amyloid, in general, we think of as a bit more of a rare disease. This is half of all heart failure, where I said there are 65 million people with it. There's probably a lot of undiagnosed amyloid that is getting lumped into this that had not been previously identified or predicted. And this just shows actual quantitation of the fibrosis as opposed to just a gross scores where we actually quantify the area in each biopsy with fibrosis. And if this is sort of the control level, you can see there are a lot of HFpEF patients who really don't look any different than normal hearts with respect to fibrosis. And then there's some that are very fibrotic. So there's room for some kind of personalized, maybe it'll be an MR scan, but if you're going to create a new antifibrotic as your answer to this disease, you got to have a large proportion of this population probably aren't going to benefit, and this is going to have to be targeted. For me, the more sort of biological Pandora's box that we opened was performing the first RNA sequencing, RNA-seq of HFpEF myocardium. So we have, as you see here, also roughly 100 patients. The controls are essentially deceased donors. They usually were in car accidents, whatever reason they had brain death, but their organs were being harvested for donation. And their hearts, I can't say they're completely normal because they weren't transplanted into somebody else, but for whatever reason, they certainly were not diseased in any way that we could make a definitive diagnosis. Yes, you have a diseased heart. So that's our control group. And then we had 33 patients with heart failure with a low ejection fraction, typical dilated cardiomyopathy. Very few of these are ischemic. These come to us from University of Pennsylvania collaboration. And then 41 of the patients from Hopkins who have HFpEF. And this was very well characterized, sick population. I think 75 or 80% of them had been hospitalized for acute pulmonary edema requiring diuresis within the past year. And we're going to get right heart biopsies from all of these folks. And we're going to do our RNA-seq, and then we're going to see what we get. So before I show the picture, you think back to the camel. And, here's a hypothesis. There will be a unique sort of mRNA transcriptome signature that defines HFpEF that'll be different from other things, given that camel. And some people would have said no. I mean, that's ridiculous. This is it's a clearly really heterogeneous disease. It's going to be all over the map. You're going to have some people here, some people there. In fact, when we first submitted this paper to a somewhat different journal, that was the criticism from the reviewers, was this can't be. Everyone knows this is heterogeneous. You must have done something wrong. But this is what the data looks like. So this is typical principal component analysis. And the HFpEF patients are all up here in the sort of bluish color. Here are HFrEF patients, and here are the control patients. And in fact, they segregate quite nicely and distinctly in groups. And there's no more heterogeneity, arguably, in terms of the overall variance of this HFpEF cohort sitting here than there frankly is in the donors. I mean, in terms of variance, that's not what we're seeing. Now, this is unadjusted. We also did an adjustment where we included information, basically what was their sex, their age, body mass index, which we expected to be somewhat different because our patients with HFpEF are definitely heavier, estimated GFR, and diabetes. So these were the prominent comorbidities that we could see in our data set clinically were somewhat different between the groups. And we wanted to adjust for that and now see what we get. And it rotates it. It's not quite as separate, although HFpEF and HFrEF are still pretty separate, and the controls are up here. So you can adjust for all of that, and you still end up with a group of patients with a somewhat unique transcriptomic signature. So that was kind of interesting. So then we looked more, what are the HFpEF patients' hearts doing? So these are all the significant down-regulated genes between HFpEF patients and the controls. We're just looking at those two groups. And these are things that are down. And it's an interesting list. This is not kind of the top of most, my thinking. I don't know about most people, but a lot of this had to do with cytosolic transportive proteins, protein processing, regulation, Golgi, stuff that related to growth. You would need to make things, and you have to, but clearly the ability of the cell to sort of move stuff around and respond was abnormal. There are metabolic and catabolic processes that are downregulated, consistent with some sort of a metabolic signature that's probably important. Here's autophagy, which is another big part of protein quality control. Now these are also unadjusted. So the first thing we did is adjusted for sex, because another popular theory is that this is more common in female because there's going to be something unique going on in women as opposed to men. We don't see it. We thought we might, we didn't. And really almost nothing changes. So then we go ahead and adjust just for obesity. because it's a big, no pun intended, factor. Turns out it doesn't change anything. We adjust for all the 5 things that I mentioned before, and basically doesn't change anything. So we're looking at a unique signature in a unique population that really does suggest some novel targets in terms of what's going on in the myocardium that may be downregulated and abnormal that we can't just explain away by certain common clinical comorbidities. Now it gets interesting when you flip it around. Now these are the things that are upregulated. And what was upregulated in HFpEF turned out to be largely mitochondrial OXPHOS genes associated with oxidative phosphorylation ATP synthesis. Although I'm not showing it here, just for everyone to know, it's exactly the opposite in HFrEF. This has been long known that in heart failure with reduced ejection fraction, dilated cardiomyopathy, these same signaling processes at the gene level and protein level are downregulated, and that the failing heart with a low EF actually is energy compromised and struggles to produce enough ATP, and its mitochondria are definitely dysfunctional. These hearts are actually kind of going in the opposite direction. This is one of the few sets of genes where things are literally in the opposite direction. So what happens if we adjust for sex here? Nothing, doesn't change anything. So it's not a male-female thing again. But here's the cool surprise. We adjust for BMI, and most of it disappears. And we adjust for all the 5 covariates, and all of it disappears. So it means that we can basically, by disappears, what it means is that once you've made a correction of your read counts for these covariates, the variance associated with the difference in the read counts is no longer statistically significant. Basically, it's like a multi-regression model. So there's something about being very heavy, and that's most of it, is the BMI, that is modifying energetics and mitochondrial gene transcription and underlying control, probably of energy homeostasis. That is, I think, a key element now that we have to go after in these HFpEF patients as their BMIs have been getting higher and higher. Next, we looked at this to slice and dice it within HFpEF only. So can we use the transcriptome, no clinical input into this, basically.
يلا بابا امسك عشان اخد this one I'll remove my coat.
It's the mic. Okay, so we tried to break this into subgroups, and we used what's called NMF factorization, which is sort of a autocorrelation of just the transcriptomes. There are a couple of thousand genes that were identified from the most significant, and we tried to ask it to cluster the HFpEF population into groups based on their gene expression, and it identified essentially 3 groups for us. We call them group 1, 2, and 3, and these are basically showing the increased correlations within those groups, and this is the number of patients in each group. And we did principal component analysis of these groups, and sure enough, it turns out that group 1 is the closest to heart failure with a low ejection fraction, whereas group 2, sitting over here, is the furthest from this. And I'm going to show you a couple of clinical features that are really quite interesting. And this is sort of the mixed group that is mixed. It's sort of sandwiched in between. So what are these groups? Again, we didn't give the algorithm any clinical information whatsoever. They're just genes. Well, for one thing, if you're in group 1, which was the one that was closest to HFrEF, you have the highest mortality. So just based on our gene transcriptome, we can actually have a prediction of who's more likely to die. It turns out the signatures, the molecular signatures that go along with these groups are also very different. Group 1, again, the one that's closest to heart failure with a low ejection fraction, have signatures sort of what you might expect. HIPPO is related to growth, MAP kinase signaling, stress-activated pathways, cytoskeleton, remodeling, hypertrophic cardiomyopathy. These are looking more and more like what we kind of think of as heart failure. What about group 2? Group 2, remember, we're the furthest away. Well, it's mostly metabolic and immune sort of cascades. Whereas calcium and some abnormalities in focal adhesion and protein quality control stuff, they were showing up in the mixed group. So these are different signatures which have clinical implications. And we also looked at sort of general properties of these patients. Group 1 tended to be more male. Group 2 was entirely female. Their NT-proBNPs in the group that look more like HFrEF are the highest. And maybe they would benefit from drugs essentially like Entresto, or drugs that we normally use in patients who have elevated natriuretic peptides. Whereas group 2 had very low natriuretic peptide levels. LV mass was the highest and the one that was closest to HFrEF, and lowest in group 2. Group 2 hearts were basically small, not hypertrophy, not much NT-proBNP. It's the smallest chamber volumes, and more often in females. So this is a cohort where if you were to look at their heart, you'd basically be clueless. I mean, these do not really look like abnormal hearts. And yet I can guarantee these patients were just as frequently hospitalized, had just the same they had similar body mass index. They were just sort of the same clinical presentation severity as all the groups, all of them. So I thought this is sort of an interesting way of dealing with this. Not using clinomics, which is what a lot of the machine learning algorithms have been doing, but going at the genes and then going backwards. This gives actually both us and pharmaceutical companies now kind of molecular targets, because we're identifying pathways and even specific proteins and things that we could potentially go after. Second piece of the story that came out around the same time, which is also interesting, is that it turns out obesity, besides changing mitochondrial gene transcription, impacts sarcomere function. So for this, what we did was we divided just the HFpEF group into 3 subgroups. And we gave them a score, basically, based on where they were in the distribution of hypertrophy, hypertension, obesity. Diabetes just got a 1 or a 0. If you had it, you didn't. And then we took the top sort of quintiles, the most hypertensive, most hypertrophic, least obese. That's group red. The most obese, least hypertrophic, least hypertensive, that's blue. And then matched both, that's green. So we create these 3 groups where we're going to be able to ask the question, what is obesity doing? Because the only difference between group red and green is that this group has obesity. They're going to be similar with respect to hypertension and LVH. And similarly, the difference between these 2 groups is going to be their hypertension and LVH, but not obesity. They're going to be similar. And this just shows that. So we matched BMI for these 2. We matched LV mass and systolic blood pressure for the outer 2. Everything else is basically the same. They all have good ejection fractions. They had this, we didn't stratify for this, but maybe not surprisingly, the ones with less obesity and more hypertension and hypertrophy had the highest NT-proBNPs, and these were lower. So this was a huge surprise. I wasn't sure what we were going to see, but this was really quite remarkable. So we take these same biopsies, we do what's called a chemical skinning procedure, a little bit of Triton, we can isolate the cell, attach it to a length and force controller, and then we can monitor sarcomere length and keep it fixed, measure tension at different steady state calciums. It's a classic sarcomere sort of protocol. So you've got steady state calcium concentrations on the x-axis, spanning all the way from what would be diastole down here to basically systole, which is up in here, to systole during exercise, probably, which is somewhere up in here. We never get out here, but we have to go to find the peak. So what do you see? Well, here's normal. This is the normal force tension, the tension calcium dependence. Here's the hypertensive LVH patients with less obesity, and they're actually not statistically different than normal. And this is the only data in the literature before this paper was from that old European work I mentioned earlier, which were mostly from these kinds of patients, and in fact didn't see any issues with systole with higher calcium. But here, for the first time, you get to see what happens with obesity. And here are obese cardiometabolic syndrome patients, diabetes basically, and notice how dramatically depressed this is. this is about as bad. I should plot this on here for you. This is what HFrEF looks like actually, with respect to sarcomere function. It's about 50%, 40-50% depressed. And these are people who are giving drugs that are negative inotropes because we think their systolic function is preserved. And it's not so shocking. It wasn't shocking to me that we're not doing so well in that respect. We have to think about this differently. And what was even more striking to me was that the mixed group, which remember are just as obese as the blue group, but they also have hypertension and hypertrophy. That doesn't kick them up. Obesity wins in this battle for the sarcomere. You're going to get the more obese you are, the more likely something has changed in your sarcomere to really reduce your ability to increase force at higher calcium. In the range at which you're sitting there doing nothing, like in the echo lab when we make our measurements, it's not that big a deal. We don't do enough stress testing in these patients, but this is really an exertional problem. And I think this is where this does become a big deal. And I'm not going to go into the more details of the data. We've now, since this was published in the last year, we've identified a variety of post-translational modifications. We can reverse part of this with phosphatases. We're figuring out with proteomics exactly what is being phosphorylated and dephosphorylated. We've already got some molecular targets. We've been trying some drugs that are actually designed to increase this to see whether it would work. It looks like it does in this group, but it doesn't do very much when we give it to this group. So this is going to be another one for sort of more personalized approaches to therapy, as opposed to just giving the same thing to everybody as HFpEF. But this is certainly one of our big surprises on how this has changed with obesity. And I call this this is new HFpEF, this was old HFpEF. And here's, this is sort of the summary shot. All I need to know is your body mass index, which is certainly not a perfect indicator of exactly what's going on with obesity. And I can predict with a pretty good small p-value for clinical data, essentially what your sarcomere is going to be capable of generating in terms of tension for the same calcium. Right? And it didn't matter whether you had or did not have diabetes. So this is not metabolic syndrome as much as it is the obesity part. So I'll show you just a couple of pieces of new data and then go to the therapy, and we'll still try to end on time. So this is what's supposed to happen in a healthy heart. Healthy hearts have tremendous substrate flexibility. And they normally use predominantly fatty acids, something around 80% of fuel is fatty acids. They don't actually use very much glucose at all compared to many other organs. They use some ketones. They can consume amino acids, so that they really are, they're very what's the right word for this? Where you'll eat anything. Yes, they're omnivoric. There you go. What about HFrEF? HFrEF actually has problems. Fatty acid uptake and oxidation is reduced. Glucose and ketone bodies really take over. And this, again, is well known and been well described because we get these hearts, they're explanted from patients, we get all the myocardium we want in the world, and it's relatively straightforward in that context to get this data. Plus, we have animal models actually that mimic this, I think, better, and we see sort of the same thing. So there was absolutely no human data in HFpEF. The only data on what is going on here in HFpEF has come from animal models of diabetes and animal models of obesity. And we don't biopsy patients who just have diabetes and no heart failure, nor do we biopsy, right? So we didn't have it, but we have these hearts. So while I'm not going to be able to give you a complete picture, I'm going to suggest at least that something is also wrong in the state of HFpEF. So this is about to get submitted, I believe. If I see this manuscript in my inbox. We've got this, I've got the figures. So here's myocardial lipid content. And we're doing, these are all the acyl carnitines, starting with not the real short ones, but which are more branch chain amino acid related, but these are sort of mid and then upper long chain, which means they've been taken up and are in the mitochondria, basically. And what you're seeing is with flower plots that sort of look like flowers, is that the control group is out here. Here's HFrEF. HFrEF, as anticipated, a lot of, there's much less fat basically in the myocardium. There are problems of fatty acid uptake and in fact utilization. That's what we've seen before. But here's HFpEF. It's in blue. And it's basically also inside the control flower. It's actually very close to HFpEF. So despite the fact that these patients have a preserved ejection fraction and they are obese, they've got lots of fat. The heart isn't taking it up. It probably isn't burning it. That's our expectation. And we've got other data in terms of proteomics, and we're still working now in some other human studies to really prove effectively one way or another what it's really doing with the fat. But this is already very much a surprise. The other thing I'd like to show you is that if you did this from blood, and this is all you can find in the published literature, this is the first heart data that will be presented, but there are blood studies with proteomics. It's kind of the opposite. Now the HFrEF people are out here. It's actually the control flower is the one that's the smaller one. And fat in blood is often elevated. And not surprisingly, this will be elevated in patients who have cardiometabolic syndrome and a lot of obesity. So I don't think we're going to figure this out from blood. I think we're going to try to figure this out from the organs that are affected. The other thing I'll show you from the gene expression analysis, if you just look at genes that are associated with fatty acid uptake and oxidation, PPAR alpha, showing up here as one of the more significantly down-regulated genes. This is exactly the picture we see in HFrEF. This is not what we were predicting. This is not what you see with obesity, at least in animal models. These things are generally up, and there's a lot of fat, and fat shows up in the heart. So very, very different. So to summarize sort of what does this all mean? For one thing, it means having a preserved ejection fraction doesn't necessarily mean underlying contractile function is necessarily preserved, right? This is a chamber kind of property. EF, we know as clinicians, is not a really great indicator of much. And I think this data is kind of proving that in spades. And other things, including vascular load and structure and geometry, maybe keeping the EF there so they're just not dilating. But sarcomere function, I think that's a real thing. And something we need to think about differently. Furthermore, obesity and HFpEF is not just obesity, nor is it heart failure. It's its own kind of unique mix. These two things have colluded now to create a somewhat different syndrome. And I think we have to, with I think, challenged metabolic flexibility. So are we getting any closer to this? And I'll finish up. The way we treat it now, this is traditional, diuretics, hypertension, arrhythmia. This drug, which is commercially known as Entresto, showed up and has now been FDA approved for heart failure with ejection fractions up to 40%. That's actually very low for our population. Most of them were in the 60s and above, and it's not approved for that. I don't know how it is in Canada. I assume it's the same. It's approved for up to 40%. Or you have a broader range. The most recent one was an SGLT2 inhibitor. So empagliflozin, which is a diabetes drug that works by blocking this sodium glucose transporter protein, which we knew was in the kidney. It's not even supposed to be expressed in heart muscle. And yet, surprisingly, a safety trial showed an incredible signature for improved mortality and reduced rehospitalization rate. And the definitive trial was late last year, and the FDA approved it just a few months ago. And this is approved for EF greater than 40. This is our only true HFpEF approved drug. And it's a diabetes drug that no one was thinking about, until they did a safety trial. And there are a couple of other ones. I just saw a recent notice for it's the dapagliflozin, which is another SGLT2 inhibitor. It looks like they have got positive data. There'll be other ones on the market. This is our first win, right? And these are the kinds of changes that we for a while we were only dreaming about. We never finally see these kinds of primary endpoint curves show significant differences between them. For the Entresto one, it was actually borderline, and that's where the FDA more or less decided they would create this cutoff, and it's at a lower ejection fraction. For the SGLT2 inhibitor, this change is one of the biggest people have seen for anything for a while. And the p-values were not subtle. This is real, and it was real no matter how they sliced and diced individual whisker plot, subgroups. Now, one of the things about these trials, which I think of as a scientist and clinician, is how does this relate to the patients we see? Remember, I gave you that slide a long time back where I showed you this is what we have in Baltimore. Well, for these trials, the vast majority of the patients were class 2. In Baltimore, 66% of our patients were class 3 and 4. We take care of it. It's a much sicker population. These are sort of walking heart failure patients. The median BMI was 30. In Baltimore, it's 41. The ejection fraction median was 57 in this trial, but a third of the patients in this SGLT2 inhibitor had EFs less than 50%. None of our patients are less than 50%. All the people we're taking care of, it's generally around 65 and rarely below 60. So, as a clinician, I'm asking myself when you read these papers, again, personalized medicine, are these are the people I'm taking care of, and what's the data for that? And it is a little problematic. For example, just with ejection fraction, this drug, this combination of an ARB and a leprolysin inhibitor seem to work better the lower the ejection fraction. And I don't know what it's going to do actually in EFs that are 65 and up. This was simply median, greater than 57. We didn't have that many patients. And then with the SGLT2 inhibitor, the whisker plots are also drifting up, where the higher the ejection fraction, the less the effect. It seems to be doing its best job, and it's already approved for HFrEF. So we still don't really know exactly. We don't know how these things are working exactly. And it may be these are our group 1 patients that are kind of driving, the ones that already look more like HFrEF to begin with. But that means we still have 75% of the HFpEF population where we're probably just scratching the surface. But that's my guess. Okay. Last couple of ongoing trials. This, I'm very excited to see what happens. This is going after obesity. So the Novo Nordisk drug, semaglutide, is a GLP-1, glucagon-like peptide 1 agonist, and the STEP-HFpEF trial is ongoing, and we're really going to get a sense as to whether or not we reduce weight. It'll help these people. Pirfenidone is an antifibrotic. There was a Nature Medicine paper that was just published recently. The effects were mostly on MR-based metric. I can't say they were really overwhelming. Sorry, Luigi, who actually works in my place, who I think came up with this idea and is moving it forward, but we'll see. And again, I showed you, not a lot of the patients actually really have that much fibrosis, I think. So this one is still active. There are a whole bunch on this list before. When I went over and looked at them before coming here, most of them have failed, so they're no longer on the active list. Ranolazine is a late sodium inward current blocker, so it's antiarrhythmic, which may have some mitochondrial effects on fat metabolism, although I'm not sure it's in the right direction. And I'm going to just leave you with the one that we're pursuing, and I'm going to talk about at the Lucian talk on Friday, phosphodiesterase type 9, because I'm suspecting most of you don't know what this is. It's a phosphodiesterase. So type 9 turns out to be the other of the 11 PDE superfamily that regulates cyclic GMP. PDE5, which is well known, is the target of the drugs like Viagra, sildenafil. PDE9, we showed a few years back, is specific for natriuretic peptide-associated cyclic GMP, not this. And we don't have a name for it because it's still in basically clinical trials now, but this was an experimental drug. First developed for neurocognitive diseases, it's in the brain. Most of that work has not been successful. There's a lot going on right now in heart. And I'll just show you one quick panel from a paper we published now, I guess 6-7 years ago, where we compared, just to show that this natriuretic peptide thing is real, a PDE9 inhibitor in red and a PDE5 inhibitor, sildenafil, in blue, in a mouse model of pressure overload. When we first apply the pressure overload in week 1, and we get disease, we get reduced fractional shortening, we get dilation of the heart, we get increased hypertrophy of the heart. And then at this point, the drugs were put in their oral food. And what you can appreciate then is both of these drugs are working very nicely to reduce chamber size, to improve fractional shortening, and reduce left ventricular hypertrophy. But when we add a nitric oxide synthase inhibitor, pill name, to the drinking water, we now block nitric oxide pathway. That's the cyclic G PDE5 regulates. And lo and behold, PDE5 inhibition, which is blue, doesn't work anymore. Doesn't reduce chamber size, doesn't reduce LV mass, doesn't improve fractional shortening, but the PDE9 inhibitor still does. And this was the first paper to really demonstrate this dichotomy. And it was partially for this reason that we tested this out, particularly in a postmenopausal female mouse model. So this is the paper I'll talk about Friday, but I want to just give you one quick cartoon from it, because I think this is going to move forward also in a clinical trial. Basically, these are very obese, overectomized female mice. This is my BMI 41 mouse. And those that ended up on a placebo as opposed to a PDE9 inhibitor, there was a significant reduction in weight, in obesity. And it turns out they basically, and this is just shown here, overall body weight went up, and the placebo group generally was the same or trending down in the PDE9 inhibitor group. This was not because of lean mass, it was all fat mass, it's mostly in their abdomen, and it was not because they stopped eating. This is not an appetite suppressant, which is what most of our obesity drugs are. And it was not because their total activity changed either. So as I say, I'll discuss this much more Friday, but there's a very interesting story of how this is working and where it's going. So I'll end with this idea that it's not just hypertension, hypertrophy, or diastolic disease. With this intersection with morbid obesity, we've got a very different pathophysiology and new avenues. And these treatments are not really targeting this. I mean, the first one, as I say, is a metabolic drug for diabetes. I think precision-guided therapy based on subset identification is both going to be important and is feasible. And with that, I'll stop. This is the team at Hopkins. I've mentioned Kavita, who set up the heart failure HFpEF clinic. Some of the folks along the way, you'll hear more about Sunita on Friday. We've got collaborators at Amgen, UPenn, and Cedars-Sinai, who are doing basically proteomics, metabolomics, all sorts of other omics, and work with us. And I'll stop there and say thank you. And hopefully,
if you stop sharing the screen, you can see in the chat whether we have some questions. So thank you very much, Dr. Kass. That was really a wonderful talk and very clear, even for the individuals that are not necessarily cardiovascular experts. Well, it certainly worked. We've gone beyond time for reasons that have nothing to do with you, but our own ineptitude. I'm looking to see if we have questions in the chat. So there are, I will ask, one of them, does sarcomere contractility change with weight loss? Have you biopsied and looked temporarily?
Yeah, fantastic question. We want to do this. I've been angling for some sort of probably, first it was going to have to be bariatric surgery patients, because that's the only way I knew getting the weight loss. Now that we have the GLP-1 agonist trial, that's probably the way I'd like to go. And that's something that we are trying to get going at Hopkins, because we're the ones who will get the biopsies. You know, obviously, if it works, if we can do that, you move further down on the x-axis, and indeed your tension rises on the y-axis. That would be really brilliant. It'd be a great discovery, but no one knows that yet. It's a great idea.
So another question was, has your group studied or thought of alternative hypotheses to explain the decrease in acyl carnitines in the myocardium of patients with HFpEF, such as cofactors, enzymes, and transporters necessary for myocardial function, in situ carnitine, alpha lipoic acid, thiamine, and some of them be
Right, this is also a really, really great question. The answer is yes, and it's still sort of work in progress. I mean, part of our problem is that we're dealing with a 3 milligram piece of frozen myocardium that's been taken at a moment, and we can only take these kind of snapshot pictures. Even the metabolomics work, this doesn't really give us flux information. It doesn't tell us where the bottleneck is. We don't know. We've measured genes, and I've shown you the genes. We're just getting the proteome. Some of the proteins that are involved very much in fatty acid uptake, for example, are downregulated. I mean, they're lower levels of expression. And I don't think it's going to be just one thing, but we're very keenly interested to figuring this out. And there are some clinical data that we are planning on doing now. I think we're hiring the person to help to do this. There's a PET scan idea where we could measure essentially fatty acid uptake and oxidation with a PET tracer, where we want to take HFpEF patients versus obese patients, but no heart failure, and compare what their hearts do, basically, both at rest and with exercise, how much fat is actually being taken up and then ideally metabolized. We're going to do, I think, the same sort of cath lab protocol that you may have seen Zoltarani, whose picture is still on the slide over here in the far right. He published in Science about 2 years ago, first time in human HFrEF, where they had cannulated the coronary sinus, and they had a peripheral arterial and venous line, and they basically looked at what fuels are coming into the heart and out of the heart. We want to do this in HFpEF. So we're going to be doing that, I think, at Hopkins in our cath lab. And my ultimate goal with that study is actually to then give the patient C13 labeled comitate and follow the carbon and find out precisely which species, doing essentially then targeted labeled carbon metabolomics. Where is it getting stuck? Measuring the flux of these things is difficult, because even with fresh tissue, we don't have that much. And these assays are going to be somewhat hard to do. But these are the kinds of things that we want to do, because I think, as you're suggesting, it'd be really important to figure out where the problem is, because that's going to be the target, actually, from a therapeutic standpoint.
Very good. Thank you very much.
Sure.
It seems like we don't have any further questions.
What question
You do?
Was this bulk or single cell RNA?
This was bulk. We're now doing single cell with a collaborator at the Broad. So by, I don't know, October or something, we'll have a major data dump. We'll sort of see where. There too, we had to do a lot of prior prep work, because it had not been done with samples of this size. And we're not going to get as broad a coverage. We'll probably end up with maybe 2,500 to 3,500 different genes, things that we'll be able to pick up compared to what you could pick up if you had, more. But I think we'll learn something.
And the genes that you were able to see, was this like from one purified cell population, or this is from all the cells that you got from that?
Everything, no, there's no, it's just ground up RNA from the entire, so it's coming from everything. Could be, it's vascular, it's all cell types. It's just bulk myocardium. Right. Sure.
Very good. Thank you very much, all. Thank you again for a
Thanks.
fantastic talk.
Thank
and
you.
we look forward to hearing more of your work in the course of the week.
And you will.
Thank you. Bye now.